So now that we are learning a little bit about how gametes are made, let's also think about, well, what happens to the chromosomes during this process, and what about the genes and alleles? So let's now even simplify this even further, where we're going to look at just one pair of chromosomes. So we're looking at chromosome pair 16. And let's assume that somewhere along this chromosome there is a gene, and this gene codes for freckles. Okay? And so we're going to highlight this with this yellow bar here, and we're going to put a big F for freckles, which is dominant, and little f for not having freckles, which is recessive. And we know that prior to producing gametes, that the amount of DNA always gets doubled. Okay, we'll talk a little bit more about this in just a moment, but it becomes duplicated. And so now, instead of just having one big F amount of DNA, or one chromosome here of chromosome 16, there's actually been duplicated. We have double the amount of DNA. And so you can see now there's actually two places where we have this allele for the big F from the gene for freckles. And same situation over here for the blue chromosome. Now, what would need to happen then in order to produce gametes that are only going to have one chromosome? Well, you might be thinking, oh, well, it's got to divide once and then it's going to divide another time, and that would be correct. So in the first cell division, you end up putting all of the pink DNA material, right, this part of the chromosome, over into one cell, and you get the blues coming down this way. Right? And then you say, okay, well, now it's going to divide one more time. And so to divide it, what we need to do is get rid of this little connection area right in the middle and separate those duplicated chromosomes so that now they come up here. Now we end up with a cell that has just one chromosome. Instead of having a paired chromosome like back at the beginning, it now only has one chromosome. So we've gone once again from that diploid situation to a haploid situation. And here's the other one, and then same thing for the blues, right? Here's one of the blues, and here's the other one. And that's, hopefully you were kind of seeing that as we were talking about this. Now, let's actually add, though, the correct grammar and terminology to these things. So it turns out that over here we're actually in what we call interphase. So it's before the cell has even started to go through meiosis. And in interphase, that is where the homologous chromosomes are duplicated. Now, it's important that we recognize that while they are still connected together, it still is just considered one chromosome. So even though it has double the amount of DNA, it still is only one chromosome. So this is still just a diploid cell. It's still only here, we only count this as two chromosomes. Now, after we go through this initial step, then we come over, and the second step is that homologous chromosomes separate. So notice that we said, the pinks went up here and the blues came down here. This is important. And at this moment, now these cells are haploid. So this is a haploid cell, even though this is only one chromosome with double the amount of DNA, but it's only got one chromosome because they're connected together. And this all occurs, this process of separating homologous chromosomes occurs during the first division of meiosis, called meiosis 1. Then you go through a second division called meiosis 2. And in meiosis 2, sister chromatids separate. So we actually now have a name for this duplicated DNA. Each of these are called sister chromatids. So when the homologous pair of chromosomes were duplicated, each side of that chromosome now becomes called a sister chromatid. And then at the moment that these sister chromatids separate, we can now count each of those sister chromatids become their own chromosome. And so that is why now this cell continues to be haploid, this cell continues to be haploid, and so forth. This is the process of meiosis. So here's a more detailed view of meiosis. And there are names to the different phases within meiosis. So first of all, notice that we are here in meiosis 1, and the chromosomes have already been duplicated because that occurs prior to meiosis happening. And the first phase is called prophase. And this is a time also when the duplicated chromosomes, so the sister chromatids here, from each homologous chromosome pair, come together and form a tetrad, where the chromosomes actually cross over and then swap tips. So now you end up with, in this case, red chromosomes with blue tips and blue chromosomes with red tips. In metaphase, the microtubules attach to that center portion of the chromosomes called the centromere, and they start to tug at the chromosomes, and they line them up, such that the homologous pairs of chromosomes are lined up such that one homologous pair will go one direction and the one will go the other direction. So the homologous pairs are going to be separated. And that happens during anaphase, right? Notice that the sister chromatids, though, remain attached. So the pairs of homologous chromosomes split up, and then the cell goes through telophase and cytokinesis, where now the cell actually divides into two haploid cells. Then in meiosis 2, you once again go through a prophase, where you start to form your mitotic spindles. They attach to the centromeres. They start to tug and pull, but in this case, they're going to pull apart the sister chromatids. So sister chromatids separate. Then you go through telophase and cytokinesis, and you end up with 4 daughter cells from that initial diploid cell. Now, because this happens where the chromosomes are lining up in meiosis 1 with the homologous chromosomes ready to be separated, they can line up, though, in any way. You can have, for example, possibility 1 with the blue chromosome on one side and the red chromosome on the other side, and the small blue with the small red. But you can also have this as being big blue, big red, little red, big blue. So the way that these line up is independent of any factor, and so it's random how these things are going to line up. And as you follow that through the first cell division of meiosis 1 and then the second cell division of meiosis 2, you end up with different possible combinations. So in the case of this chromosome, where we're looking at 2 chromosome pairs, there's a combination A possibility, a combination B possibility, a combination C possibility, and a combination D possibility. Now this is for an organism that only has 2 pairs of chromosomes. But how many pairs of chromosomes do humans have? They have 23, right? So when you only have 2 pairs, it's a 2 to the 2 problem. When you have 23, it's a 2 to the 23 problem, which is 8,388,606. So that's the total number of different types of eggs and sperm that humans can produce. Now actually, the number actually gets larger, because if you recall, in prophase of meiosis 1, the tetrad is formed, and you have these crossing over sites, or these chiasma, these sites of crossing over. And so you end up with chromosomes that are blue with red tips and red with blue tips. So there's actually even these other additional possible gametic combinations, and we call these recombinant chromosomes, where they're, half came from the maternal grandparent and half came from the maternal grandfather and maternal grandmother, and they got switched up during the crossing over, and so you end up with additional combinations. So actually, there's many, many more possible gametes than just the 8 million. There's way more than that, and this is the reason why two human, a male and a female human, never can produce identical offspring from 2 independent fertilization events. It always happens. You only can get identical offspring from 1 fertilization event, and then that initial egg and sperm that came together to form a zygote then separates into 2, and that's what you call identical twins. So that's kind of an interesting thing to think about. So anyway, this is an introduction to the process of making gametes, the process of meiosis, which will help us figure out if Jack and Jill should get married or shouldn't they get married.